I'd like to thank everyone for joining us to this APDA Passport to Research series. My name is Dave Grosby. I've been a sportscaster in the Seattle area for about 35 years. In October of 2018, I was diagnosed with Parkinson's disease. So I've had it for a couple of years now. I was fortunate to have a connection with APDA, and I'm very happy to be involved in this series. APDA has a dual mission, providing help for those today living with Parkinson's and hope for tomorrow by investing in critical research products, which is why I'm proud to be associated with this organization. Research is what we're here to talk about today. APDA has invested nearly $51 million into critical research since 1961. Northwest Chapter is proud to be the sole funder of the Parkinson's Disease Registry, which helps speed up research process by connecting researchers to willing participants. Make sure you keep an eye on your post-event email for more information about that registry and how you can get involved. There is some amazing research going on right here in the Northwest. We do hope through this series, you'll learn more about the work being done, the people behind it, and how you can be involved. Before I introduce today's speaker, I want to thank the sponsors of the program. Gold sponsors Kiowa Kirin, Synovia, and Niagara Bottling. Silver sponsor Amneal, and bronze sponsor Boston Scientific. Their support of APDA and the value of the educational programs like this is immensely appreciated. Today's Passport to Research series is entitled Novel Trials with Novel Mechanisms. Again, I encourage you to ask questions in the chat or in the Q&A, and we will be sure to get to them. So let's begin. Our guest, Dr. Jason Aldred, is a board-certified neurologist and fellow of the American Academy of Neurology. He completed neurological residency at the Oregon Health and Science University, serving as neurology chief resident, then completed an additional 2-year clinical research fellowship in Parkinson's disease and movement disorders at the Parkinson's Disease Research Educational and Clinical Center, a collaborative program between the Portland VA and OHSU. Dr. Aldred conducts clinical trials for a variety of neurological disorders through funding sponsored by the National Institutes of Health, Patient-Centered Outcome Research Initiative, as well as other sources. He provides regional and national education for other neurologists and patients living with neurological conditions. He brings cutting-edge clinical trials to the region. Dr. Aldred is located in the Spokane area, Selkirk Neurology, and Inland Northwest Research. And Doctor, thanks very much for being with us today.
Yeah, Dave, thanks for having me, and thanks to APDA for what they do.
Why did you pick Spokane as your place to do your work?
Well, I did my residency and fellowship out in Portland, and really loved being in the Northwest, and was in the Midwest for a few years after fellowship. And my wife and I decided we really wanted to move back to the Northwest, and we're looking at options, and love the coastal area, but really took a look at Spokane and kind of realized that this is a really decent sized population center we didn't know a lot about, and it seemed like there was a great need for clinical care in movement disorders and Parkinson's work. And so after exploring that a bit further, found that actually there was a good foundation here. Dr. Jonathan Carlson, one of my friends and colleagues from residency, has a DBS program out here that had been well developed, and Jamie Marr, one of my now collaborators, our nurse practitioner at our clinic, was out here. So really, there were the bones for the foundation of building an even better program and more robust program. So thought it would just be a really great fit, and being somewhere where the services are needed, I thought was really important to me. And we've really just fallen in love with Spokane since then.
We're here today to talk about innovative new novel therapies being studied in PD. How much work is being done in this category?
A lot, to say simply, which is encouraging. There is a lot of work being done for novel symptomatic therapies. In other words, ways to look to treat symptoms better and more safely. And also novel therapies looking at slowing disease progression that are a bit geared differently than the past. Looking at targeting, for example, the alpha-synuclein abnormalities in Parkinson's disease, genetic abnormalities in Parkinson's disease, and then kind of building on from that.
What can you tell us about a trial or two that fits in this category, and what are you hoping to find?
Yeah, well, there's a couple of really exciting trials to tell you about, particularly that are active here at our site. There's a trial called, it's a Biogen-sponsored trial on a compound called BIB 094. And this is a placebo-controlled randomized clinical trial. it's an early phase of development, phase 1. And really what this is based on is we know that Parkinson's disease may have some involvement with too much LRRK2 protein being expressed in the brain. In fact, there's one of the more common genes, the LRRK2 gene, is abnormal in Parkinson's disease. And we see that even in people without the gene, they have abnormal levels of LRRK2, though not as nearly as abnormal as if you have the gene abnormality. And so what this trial is focused on is injecting a novel compound into the spinal fluid and like via lumbar puncture type treatment, with the hopes that it might reduce the body's genetic instructions to express and send out too much LRRK2, and hopes that it might slow the progression of Parkinson's disease and help the symptoms.
I know one of the trials you originally set to speak about today was stopped because preliminary data was not showing what they hoped to find. How common is this, and why is failure actually important in the research field?
Yeah, I mean, I think we're all taught in many avenues of life not to be afraid of failure, although it's certainly disappointing and can be discouraging. But we've been very involved in the Biogen Spark trial, which was stopped after about 3 years, and after all the patients completed the term of the trial that was designed. And this therapy, it's called cinpanemab, did not achieve what's considered the proof of concept to slow the progression of Parkinson's. And so while that's considered a failure, I would say that so far to date, all trials concerning slowing the progression of Parkinson's have been failures. So it's 100% rate of failure for disease modifying therapies, we call it. And this is therapy is focused on a monoclonal antibody, which is an antibody kind of like what our bodies make to fight other diseases. It's an antibody made by our white blood cells. In this case, it's a synthetic antibody rather than geared to fight a disease or an invader. It's designed to bind to abnormal presence of alpha-synuclein, which is there's alpha-synuclein occurs normally in the brain, but in Parkinson's, we know that it's abnormally folded together and clumped together. And so that trial was discontinued due to failing to meet its endpoint. But we did learn that it appeared to be safe. And so I can think that gives hope to other variations, perhaps, of monoclonal antibodies, that trials for Parkinson's disease. And in fact, we've got 2 other ones going on right now. We have an AstraZeneca trial looking at a compound called MEDI1341. This is phase 1 and is an antibody to alpha-synuclein. And also the PADOVA trial, which is sponsored by Roche, looking at prasinezumab as a monoclonal antibody to alpha-synuclein. So, there's still other efforts, and we hope that we may find, continued demonstration that they're safe and well tolerated, and hopefully ultimately slowing progression of Parkinson's and helping the symptoms over time. It's not been demonstrated yet.
We encourage people to ask questions, and we got one here from Bob Dixon. It is, if a person participates in a clinical trial for a new medication, has positive results, the symptoms improve during that trial, are they automatically allowed to continue with that medication post-trial? Or are they, quote, body washed of the medication while FDA approval is pending, and then hope for prescription once the medication is approved?
Yeah, that's an excellent question. I would say in phase 1 and phase 2 trials, which are the 2 earliest phases out of 3 phases of clinical trials before approval, in the early phase, phase 1 and phase 2, you're discontinued from the medication in most cases and washed out. There are some phase 2 trials that allow what's called an open label extension, where the patient might be, if they're on placebo for the first portion, they may be allowed to continue for a longer period where everyone gets the drug. Of course, remember, these are experimental therapies. They're not approved therapies. So experimental therapies, the whole point is to look at safety and benefits. So you're not guaranteed that we know enough about it that you're going to definitely get a benefit. But that would be a situation where it would continue. Phase 3 studies are very much the same way, with the exception that phase 3 often leads up to approval of the drug if that drug is shown to be safe and effective in the sense that it reaches its predetermined goals for a positive outcome. And so that would be the situation in which it's possible a person in a clinical trial would be able to continue on it if it was enabled, if they were allowed to have an open label extension up until the point of clinical approval. We've certainly seen that with other Parkinson's therapies and other treatment options and different avenues of medicine. So that's kind of how that would happen. It's the sponsors that run these trials, that sponsor trials, they're not obligated to do that, but there's a variety of reasons to do that, even after they've met the endpoint where they're looking at did they go to 12 weeks or was it at a year, et cetera, where they determined they were going to stop the measurements. And the value of having this open label extension phase out further, perhaps 6 months, a year, maybe longer, is to gather more information about safety under the guise of the clinical trial, where there's a structure and it's standardized measurements all the way through.
Is there anything in general you would want a patient to know ahead of time before participating in the clinical trial?
Yeah, I think a big, maybe I would just speak to a misconception that I see sometimes in patients that are otherwise interested in clinical trials, which they think that if they are on quote unquote like a placebo, that they have to not be treated or they have to forego treatment in order to be in a clinical trial. And I would say, first of all, the first priority is to always treat the patient for their medical conditions first appropriately. And clinical trials are never meant to encourage or have someone have a substandard of care. So the clinical trials are always in addition to the usual standard of care for ethical and other reasons. And in reality, we have things that are treating symptoms. And so we want to have our clinical trials, do need to be representative of kind of the real world to some extent. So, for example, if a experimental therapy is looking at, perhaps slowing the progression of Parkinson's, well, a majority of people with some type of bothersome symptoms of Parkinson's are gonna also be on symptomatic therapy. So, there's a common sense element that hopefully, a lot of folks will get, where they understand that this is not meant to be, one versus the other treatment versus you have to go without in order to participate. So that's a common thing that I think I'd like for patients to know ahead of time. And another thing that I think it's really important is to impress, how much safety and monitoring is going on during clinical trials. The common thing I'll hear from patients, sometimes it's just joking, but they say, well, I don't know if I want to be a guinea pig. I would actually say that even in animal research, there's a lot of safety things being geared towards animal safety in preclinical studies, and in human studies it's even more so. It's safety all the way phase 1, dramatically phase 2, and even in phase 3. And the whole point of phase 4 studies after approval of the drug is to get more information about ongoing safety over a very, very long period of time. So that's really kind of one of the things that is very emphasized in clinical research, safety.
You may have touched on this, but what is the experience like for a patient who does participate in research?
Well, it varies a lot based on the type of trial they're involved in, and the nature of the treatment, how much we know about it, our experience with it. Some treatments may be slight changes of other treatments already available out there, experimental therapies. But in general, I would break it up into early phase research, where we're looking at safety primarily, not really looking at does it benefit the patient yet, but first focused on safety. And those studies are very short. They tend to be weeks to maybe months. They may include overnight stays at a research center with monitoring their heart, doing frequent blood draws, doing frequent examinations, et cetera, to lab tests to look at safety, like in great detail. And so, but they're not very long, but there's kind of time intensive. And then phase 2 are kind of in between that and phase 3, which is phase 3 would be somewhat less frequent visits, perhaps every a few weeks in the first month or two of a study, then maybe every month or maybe every 2 to 3 months, just a research visit that would be kind of lengthy in the sense of being an hour or two longer than a regular clinic visit, but less intensive. So in general, phase 1, shorter, more intensive in terms of time, all the way to phase 3, which are longer studies, less time intensive with each visit.
Barbara asked a good question. What makes an approach novel?
Yeah, absolutely. So let me give you an example of that I think rings with just about everybody that's read about Parkinson's. Is levodopa is phenomenal for Parkinson's, right? We can replace dopamine. There's not many conditions in medicine where you can replace a chemical that's not there and make someone better. It's type 1 diabetes, thyroid disease, Parkinson's. Those are the common ones that I can think of. Knowing that, a lot of focus of therapies have been on how do we make dopamine work better? How do we make it last longer, et cetera, et cetera. So things that are geared towards dopamine replacement or dopamine stability, they're not novel, but that's kind of along the lines of the dopamine problem in Parkinson's. Something that's novel if it's the method of delivery is truly unique, let's say, or the chemical compound and how it works is very unique and first time it's ever been done. So something like that. Like for example, and I'll use levodopa because I've just kind of said it wasn't as novel, but there's a study going on right now, which is an AbbVie study, looking at ABBV 951, where carbidopa levodopa in a liquid form is being administered via a pump. And that's part of the study protocol. So it's the same old thing in the sense of it being levodopa, which is really tremendous, very effective drug, but being delivered in a continuous infusion therapy into the skin, not into the stomach or anything like that, but in a way that's really been very challenging to deliver. And levodopa, for all of its wonders that it can do, the stability of it and keeping the blood levels even is a real challenge. And so that's an example of a dopamine kind of treatment that's been that we're looking at a very novel way of delivering it, a new way of delivering it.
What adjustments have you had to make, if any, during the pandemic?
The pandemic's been a struggle. I mean, clinically, and on the research end as well. And in both cases, we're very fortunate in that we're kind of a smaller operation, which has its pros and cons. But in our situation, we were able to make changes very quickly. So we had clinic mask mandates, and long before the state mask mandates were made, and we were able to really screen people very well and keep the in-clinic visits to an absolute minimum, go to telehealth, which we feel was responsible for why we've really had very minimal COVID-related outbreaks in the clinic, et cetera, and none in the research center. So really that's been I think crucial, trying to implement measures early on the clinical side to do as much that could be done reasonably with telemedicine for as appropriate, and we scaled back as measures were slightly relaxed, and then when there was another surge, we kind of went back to that. So being able to respond to that and quickly, I think, and very thoroughly was helpful.
Doctor, can you talk a little bit about the work you've done in deep brain stimulation?
Yeah, so we've been very active in deep brain stimulation. In fact, the first deep brain stimulation case in the state of Washington was done in October of 1997 in Spokane. A lot of people don't know that, but that was the case over at Deaconess Hospital. And so since then, the program in the area has been very robust. We've got a DBS committee that meets from the different areas in the city, and we're very collaborative. So our research on deep brain stimulation has focused on a couple of things. Right now, we're primarily focused on using imaging techniques to assist in the programming of deep brain stimulation. There's a Boston Scientific poster, in fact, that is just getting presented here in the American Academy of Neurology here shortly, looking at their system to utilize programming on the tablet that's mapped to a DBS to the DBS lead for the individual patient. And one of the problems with DBS programming is you can have the patient selected appropriately and then implanted and then adjusted, but it may be that a movement disorders neurologist isn't available, or perhaps they are, and they only have so much time. And so anything we can do to more rapidly and accurately program patients with an anatomical aid, in addition to the other ways that we're doing that, is really helpful. Research that we've done locally here, my colleague, Dr. Jonathan Carlson, is a very skilled, gifted neurosurgeon. Again, I worked with at OHSU, and he's focused quite a bit on end of life kind of care in Parkinson's, and then looking at what do we do in the in the ethical dilemma of a patient who is in very advanced Parkinson's, perhaps near the end of life, and needs a DBS replacement. And so we've looked at querying kind of what patients' preferences are and what, what our experience as a center has been. We've published on that as well.
Are there things in general you're looking for in a candidate for deep brain stimulation?
Yeah, I think it's very important that for most patients for the majority of patients with deep brain stimulation, that they have had a reasonably good response to levodopa, but the medicine's wearing off. That's really kind of one of the key points. Now, there are some tricks there where they have perhaps the medicine's helpful, but not responding. The tremor doesn't respond, things like that. And overall, they are in good health with respect to their cognition, attention, concentration, memory, are fairly good health there. And they have reasonable expectations for what the surgery can provide. The surgery, deep brain stimulation surgery, is to treat symptoms of Parkinson's. It doesn't slow it down. And it's meant to complement our medication therapy, not really to replace that. So you kind of have the idea of medications and the surgery both working better together than either one by themselves. And so we kind of look for that patient will do well with that type of therapy.
Where are we in the field of advanced medication management for movement disorders?
Well, Parkinson's management right now, the standard of care is kind of, for better or for worse, medication polypharmacy, which, the idea of taking many medications to treat a medical problem is, most reasonable people would want to run from that. But in Parkinson's disease, the standard of care is still currently to use medications that we think are going to give the best benefit with the fewest side effects and most targeted to the patient's specific symptom problem. And then perhaps add on medication on top of that, so that we don't have to rely on one medication at a lower dose, but would instead perhaps rely on multiple medications at lower doses. So it seems a little counterintuitive. Most reasonable people don't want to be on a lot of meds, but in Parkinson's, it does make sense that we're using multiple meds at lower doses to try to stabilize the dopamine system rather than any one medicine at a higher dose. Now where that's really been tremendously beneficial for patients is to itemize and kind of rather individualize the therapy to the person's needs. But I think that, we're going to see, we're on the verge of some paradigm shifts with Parkinson's disease, hopefully for disease modifying therapies, although again, 100% rate of failure so far. But symptomatic therapies, I think, are getting better as well. Continuous levodopa delivery, longer acting forms of levodopa that are truly that might be fairly dramatically improved in the past. Hopefully, we may see approval of those agents that are being tested. Medicines that are like the current ones that are available, that perhaps are effective yet better tolerated at higher doses. Parkinson's management, like any other condition, you get benefits from the medication. Sometimes you have to push it to higher doses. You can get more benefit there, but you also see more side effects at a higher dose. So kind of getting medicines that are more selective, cleaner, and can be used at higher doses with fewer adverse effects. That's kind of what we're looking for and starting to perhaps see the beginnings of.
What is your favorite thing about participating in research?
Well, for me, the clinical practice of medicine informs our research needs, and the research needs are kind of what we learn from research and informs our clinical decision making on patients in some ways. I sometimes through the regimented kind of protocol, we call it, or another way to think about it as a recipe, you sort of learn a lot about the patient's condition from a different view than you would a routine medical visit. And I really appreciate that. It makes me think a little differently than just a clinician. When you're thinking more as a clinician scientist on the research end, you think a little bit differently from just the way you do just a regular clinic visit. So I feel that the two kind of inform each other, and I really complement each other quite nicely.
Barbara had another question. How common is the use of repurposing diabetic drugs in Parkinson's disease research?
Yeah, exenatide is a medication approved for, I believe, type 2 diabetes that is undergoing starting a clinical trial now. I think that one will start at our site here in the next few months, possibly. And the phase 2 data looked really promising. It was a fairly small study, but what was encouraging about that is some of the improvements in motor function, movement ability, and perhaps also cognition as well. And one of the limitations of dopamine medicines is that they're focused on the motor problem of Parkinson's and things that may have a broader, either whether it's improving symptoms, cover symptoms more broadly, or maybe slow down the progression of Parkinson's, so that it's not just the movement difficulty, but the cognitive difficulty, mood, bowel, bladder, low blood pressure, a variety of things that kind of gets at more of the core of the biochemical problem in Parkinson's is sorely needed. So perhaps we'll see something there. Again, all these trials I'm discussing, all this is experimental, nothing is approved. And other than DBS, which we kind of discussed the clinical use of that. So I like to carry a cautious optimism, even despite failures. I think a cautious optimism, looking for does this really work? Is this just hopeful, a placebo effect that from a trial that was poorly designed? So I think it's important that the trials be carefully designed and well designed, and we're looking for true benefits that can be reproduced. If any other group were to do it, you want a good science, you should be able to reproduce the benefit and not just a lark, kind of finding on its own. So that's the kind of thing I would consider.
Feel like we're making progress?
Absolutely. I mean, even with the failures, I feel like we're making great progress on understanding safety. I think the thing that's most encouraging to me, in light of not seeing any big, breakthroughs on therapeutic improvements yet, what is really encouraging is the powerful effect that medications can have that we are seeing in cells and in animal studies. So a powerful, kind of biochemical change, yet pretty robust safety profile, and that's a little different than what we've seen in the past in clinical research. Often, the potency of the therapy is kind of tied to a fairly potent adverse effect, and we're seeing, I think, the way that the experimental therapies are being pursued right now, I feel that we're seeing less of that and a little bit cleaner. And if we can just demonstrate that if these therapies can be demonstrated to actually work, really a new chapter.
Last question from me. How did you wind up getting involved in this field?
Yeah, well, initially I was planning on studying Latin and Greek, and classics was my actual focus initially, although I really like neuroscience as well. I thought about attending med school, but I ended up kind of writing my senior thesis on mental illness and neurological disorders in ancient Greece, looking at kind of how antiquity viewed what we now call neurological and psychiatric conditions. And I was just really, my interest was piqued by the idea of origins of what is consciousness, and then in the case of movement disorders, what is free will? What's the difference between learned movement, which is what's damaged in Parkinson's, and volitional, like a spur of the moment motor behavior. So I guess it all ended up sort of kind of going together and following movement disorders. I kind of came at it more from maybe a philosophical bend than a pure science, but I also had a degree in biochemistry by the time I got all done with it. So it was kind of one thing led to another, just following my interest and passion.
Novel trials with novel mechanisms. Dr. Jason Aldred, thank you very much for being with us today. We appreciated everything.
Yeah, thank you, Dave. Thanks for having me.
I want to remind everyone we've got one more of these coming up next week, and it's going to be with a panel of people who have participated in research projects. So if you're thinking about being involved, we'll have some firsthand experience for you coming up on next week's show. We thank you very much for being with us, and this will be recorded. You can watch it and follow it. Make sure you check your emails for more information. And thanks everyone for being with us today.